All right, we are group number 5, and we are doing figures 3A through D. And I'm Ryan Villacrucis, and my other team members are Kelly Kaufman, Blake Sanchez, and Diego Hasso. So what the overall question that the scientists are trying to emphasize in this experiment is, how can the survival of dopaminergic neurons be characterized by the presence of Th plus cells? And so some terms that we need to know before we actually try to answer that question is that tyrosine hydroxylase is an enzyme that catalyzes the biosynthesis reaction of dopamine. The Th plus cell is a neuronal marker for dopaminergic neurons. A neurite is an extension of a dendrite or axon outward from the cell body of a neuron. And the putamen and the caudate forms the dorsal striatum of the midbrain, which is really important because it facilitates voluntary movement. And the vehicle control group is the group that's not transplanted with the grafted cells. And the term innervate means to provide nerves to an area.
Alright, so next I'll be talking about experimental rationale. So the reason for this study was done was to find out whether human iPS cell-derived dopaminergic neurons could function and survive in primates with Parkinson's disease. Now, the study was important because iPS-derived dopaminergic neurons had only been tested for battling Parkinson's disease with rat models only. And while rat models come close to what you can find in humans, primates are a lot closer. And a lot of other studies had done work with transplanted embryonic stem cells. But a problem with that is it's been found to be a little unethical. So it's pretty important that we're starting to use this newer development of iPS-derived cells to battle diseases like Parkinson's disease, because now you're not actually destructing a human embryo, which a lot of people find politically and ethically incorrect. And so, yeah, in this, we'll be analyzing figures 3A through D, and it's going to show the survival of these derived dopaminergic neurons by looking at the quantity of Th cells present. Now, what were the results that these scientists were hoping for from the beginning stages? So if scientists could prove a high enough survival rate for these iPS-derived dopaminergic neurons in primates, it would then argue that this more ethical method of derived dopaminergic neurons should be tested on humans with Parkinson's disease. Now we're going to talk about the design and flow of this actual experiment. So Th cells were identified by histological analysis, staining them with 3,3-diaminobenzidine. The study estimated the effective range of these grafted cells by looking at the relationship between positive behavioral recovery, analyzed of what they actually saw in the primates through video footage and such, and then comparing that with the area innervated by Th fibers when they looked at them closely with microscopes. So some of the microscopes they used for this Th staining and to view what they looked like was this one shown below. And this is a fluorescence microscope, and the model they used was a BZ-X700. And so what they were doing was, when they were analyzing these areas, they were looking at the optical density of the Th cells to see how many there were, and then they'd later look at the shape and size of them. And also important note to look at is the primates were examined over a period of 24 months after the transplantation of these iPS cells. And this is important because in these figures we're analyzing how these cells are able to survive. So you really want to have a longer term experiment to see what their growth rate is like and see if their survival is good enough to be used to help fight Parkinson's disease. And so next we'll be moving on to Ryan to actually see the results of the experiment.
Okay, so for figure 3A, we have two histological data from two different monkeys concerning the spread of the Th plus cells stained by a drug called DAB. On the top row of images, we have successive before and after photos showing the left side view. So the top row images, we have histological data of our experimental primate model, while on the second row of images, we have histological data of a primate model that wasn't subject to the transplantation of dopaminergic neurons. And so on the left pair of images, we have successive photos of successive before and after photos showing the left side view of the midbrain, while on the right, the pair of images on the right show the right side view of the midbrain of each respective primate model. And so what we see in the photos for monkey number 10 on the top row is that we see a gradual spread of Th plus cells originating from the graft to its surrounding areas, while the Th plus neurons with the Th plus neurons extending its neurites into the caudate and putamen of the primate models. And this is in contrast to the vehicle monkey that didn't undergo transplantation, in which no significant indication of a spread of Th plus cells were shown. And this leads us to data table C, where the neuronal fibers of Th plus cells were examined in varying areas of the midbrain. And in this table, the graft cells, according to their origin and which side they were identified, either left or right, were quantified by how much area the Th plus neuronal fibers were taking up in the dorsal striatum in percentage points. And according to this data, dopaminergic neurons derived both from healthy and PD patients expressed similar innervation patterns in the dorsal striatum of the host primate model, with some primate models showing a total area of up to around 90% innervation, while the lowest area covered exhibited in the primate model was around 10%, indicating at least some degree of innervation for all primate models. And so, finally in table D, the morphology and size of the Th plus neurons in both the graft and the dopaminergic neurons found originally in the substantia nigra of the primate model expressed similarities with one another.
Okay, now that Ryan has told us what the data shows, what does the data mean? So in the figure 3A and B, contrasting the Th cells innervation, it showed that there was a great survival of the grafted Th and dopaminergic neurons. As said earlier in the presentation, Th is an enzyme that is a marker for dopaminergic synthesis. So we can conclude that if Th cells were present in these grafted cells and grafted striatums, then there must be dopaminergic neurons as well. So this is really good news that these grafts can survive in primate brains. And then in figure 3B, it shows that the vehicle group did not show growth of Th fibers, contrasting the grafted monkeys. So that shows us that grafting actually works in comparison with our control group. In extended figure 4A that you can see on the right side of the slide, these are all of the monkeys' brains that were grafted, contrasting the healthy donors in the top and the Parkinson's donors in the bottom, and the dark portions are where the Th cells are. Again, these are markers for those dopaminergic neurons. So you can see the dark areas especially monkey number 2, monkey number 1, number 10, and 11, you can see these grafts are really producing those TH cells, aka a marker for those dopaminergic cells. And then in figure 3C, the table that Ryan went over, you can see that this demonstrates that both healthy and Parkinson's donors had a great percentage of TH cells ranging from 10% to all the way to 90%. This really shows that we do not need a healthy donor for a successful graft. That is huge and great news, meaning that in future research and future implementation, somebody with Parkinson's disease can use their own tissue and their own cells to be grafted into their striatum to start producing these dopaminergic cells. And we would not need an outside donor to give us that tissue. Figure 3D shows the contrast of the morphology and size of the different TH cells and dopaminergic cells. Since they have similar morphology, aka similar function, morphology, similar form, this implies that they also may have a similar function producing dopamine in that striatum. So that means that these grafts and synthetic TH cells are comparable to our natural dopaminergic cells in our substantia nigra. Extended figure 4B at the top shows all of the monkeys' TH cells in high def. So on the figure monkey number 2, healthy on the figure on the left is an extended version, while the figure on the right is a zoomed up version where you can see the cell bodies of these neurons and the dendrites. And these are for all healthy patients on the top row and all of the PT donors on the bottom row.
And lastly, moving on to some critique and feedback. But first off, we'd just like to say that we support the conclusion as the experiment clearly demonstrated TH plus cell innervation, which in turn provides evidence for the survival of grafted dopaminergic neurons. But let's get into some critique from some novel cell and molecular biologists. We think they could have labeled the stained caudate head, putamen, and striatum images with more explicit labels, indicating where exactly the putamen is or the caudate head, because just looking at them from a novice standpoint, it's kind of difficult to determine what we're looking at. Also, adding color to the high magnification images would help our understanding of just focusing our attention on what's really important in the image. Some questions we have were, how did they determine how similar the grafted TH plus cells were to the substantia nigra TH cells in part D? Did they individually compare the sizes of the TH plus cells, or did they just eyeball it? They didn't really specify in the method section of the article whether they did this or not. But yeah, that is going to be all the critique we have for this article.